Today's webinar is part of Advanced Hardwood Biofuels webinar series, and all of our webinars are recorded and archived. You can find this webinar and all previous webinars. They will be up on our website. If you look at the little box in the left-hand bottom corner, there's the website link in there. So if you know of anyone who wanted to join us who couldn't, you can send them that link, or if you want to rewatch the presentation later, it'll be available to you probably the end of this week. The Advanced Hardwood Biofuels Northwest project is funded by the USDA, National Institute of Food and Agriculture, and they very graciously gave us 5 years of funding and an additional year, so we're continuing on till at least the end of August of this year. And the premise of the project is that we're trying to see if you could grow poplar trees essentially as an agriculture crop in the Pacific Northwest, and every 3 years you would harvest the trees and use those wood chips to create useful products like transportation fuels and also bio-based chemicals and bioproducts like paints, plastics. And because of the ways the trees grow, they resprout from the root mass, so it's essentially a perennial style crop. We are a consortium of university and industry partners. We're led by the University of Washington, but a number of other universities are involved, such as Oregon State University, Washington State, University of California, Davis, University of Idaho, the Agriculture Center of Excellence at Walla Walla Community College, and then we're very grateful for our industry partners, Zia Chem and Greenwood Resources. Oh, wrong slide. Here we go. So we're going to run questions at the end of the presentation, but please feel free to type in questions into the chat box as we go, and at the end I will read out those questions for Amy and Steve to answer. So at this time I'm going to pass the microphone over to Dr. Amy Klocko and Dr. Steve Strauss. Amy is the one who's going to be presenting today, but Steve is with her in the office, and they're going to be talking about genetic engineering as a tool for improving poplar feedstocks, and some of their research looking at this in the context of AHB. So I'm going to turn off my mic, and if Steve and Amy, you could turn on your mic, I will pass it off to you.
Excellent. How's the sound coming through on everybody's end? Excellent. Glad to see that. Okay, well, thank you for joining us today. Very excited to be talking with everybody and sharing some of our work and some of the background behind the project. So just for a brief overview, I'll go through some of the reasoning behind the work. Why are we interested in tree genetic improvement? What are some of the options for creating or building a better poplar tree, as it were? What are some of the regulatory and social concerns, especially in regards to the use of genetically engineered trees or some potentially exotic, potentially invasive trees? Go through a little bit of the lab data on what we know so far, just a couple tidbits to whet everybody's interest, and then go through a bit of the future prospects. So why are we doing this? So we think that genetic engineering for poplar trees in general could be a very valuable tool both for economics and for sustainability. So we know that we can use genetic engineering to improve our feedstock yield, so more biomass, growing more trees on the same amount of land. And we also know we can get a lot of very useful feedstock quality modifications. So of particular interest would be an increase in product formation. So if you can make trees that are more extractable, more fermentable, you can get a better yield of the products you're interested in from the same amount of biomass. You can also have potential input reductions, inputs being pesticides, irrigation, or other costs into the growing of poplar trees. And we also know now that you can make some very valuable co-products in the trees themselves. This would be the synthesis of chemicals, bioplastic, enzymes, or other useful materials in the trees themselves, which could then be extracted either as a separate stream or at the same time as the actual hardwood biofuel original goal of getting petroleum-based products. However, at this time there are some severe regulatory limits and marketplace restrictions and social concerns, especially for the use of genetically engineered trees. So I'll go into a little bit of that. Just wanted to say that third-party certification such as the FSC system does not currently allow for the use of genetically engineered trees at all. And also wanted to point out that all the advanced hardwood biofuels trees themselves are not genetically engineered. They're all hybrids, but not genetically engineered. And one of the reasons why there are all these concerns and restrictions is the issue of gene flow. So I'll go into a little detail on that later, but just to say this is one of the main concerns on basically having trees or their genes move from plantations to wild stands. I also wanted to point out that we know lots of information about poplar genetics. Part of this is because we actually have a complete genome sequence. First tree ever sequenced was Populus trichocarpa. This is work by a very large group of scientists. You can see here I put in a screenshot of the article title as well as the people involved in the work, so quite a large research group. And this would be a picture of the tree on the right. You can see what they look like. And I want to point out there's a lot of naturally existing genetic diversity here that researchers and poplar growers and tree breeders have been taking advantage of for many, many decades. And it's not just a single tree that we have the sequence for anymore. We've actually done many individual trees as well as other species of poplar. So we know a lot about the genetics in general. And if you do have a poplar plantation, such as one shown here, there's a lot of stress on it. So there's a lot of opportunities for improvement. So we know that the trees face a lot of problems, such as insect pests, like the one shown here on the left. They're also susceptible to possible diseases, like the poplar rust shown here on the right. That's some fungal rust growing on the leaf. You also get competition with weeds and other ground covers. And of particular interest right now, especially in light of climate change, is the idea of water-related stresses, so drought stress, or as strange as it sounds for us here this time of year, the idea of not having enough water. It can happen. We also can get more animal pests, such as the little Oregon beaver shown here. I want to mention he's our mascot here at Oregon State University, so we love the beaver, but we do not like that he eats trees. So it's a love-hate relationship. No, actually we love the beaver, and that would be more of a management issue, but just thought I'd bring that up as another possible issue that landowners might face. So if you want to build a better tree to perhaps deal with a lot of these potential stresses, what are some of the options? So there are two main routes to get a better poplar tree. So one is traditional breeding. This is where you have two parent trees of genetically distinct variety, and then you can cross-pollinate them together and get a whole vast number of offspring. And these will be different from the parents and different from each other, and they may have fantastic new traits of interest, which can then be analyzed and screened, and you can find a tree with more of the traits that you want. It's also going to incorporate a whole bunch of other traits from both parents, so you're going to get a lot of carryover, which is both good and bad. You get a lot of variety. Second option is to take advantage of genetic engineering in science to take your favorite poplar tree, shown here, and to specifically add a single trait to it. And I've glossed over it here and called it the arrow of science. I'll go in a little more detail in the next slide. But basically what you end up with is the exact same tree that you started with, but with one change to it. So you don't get the variety of offspring as you do above with the traditional breeding, but you do get one very precise change. And so this can be a way to rapidly incorporate a single trait of interest. And I do want to mention that these are complementary approaches. They're not mutually exclusive. So you would do the genetic engineering within the traditional breeding pipeline or within the background of your favorite trees obtained through traditional breeding, or your genetically engineered tree could be put into the breeding pipeline as well. So these are not two completely separate approaches. So for a little bit of more detail on genetic engineering in particular, so this is a way to very quickly and specifically add traits to trees. It can be used to add traits to any organism, but I'm just showing trees here as our example of particular interest. So what we do here in the Strauss lab, or in other genetic engineering places, is you start out with small pieces of plant tissue. So what you're looking at here is a petri dish with little circles of leaf tissue. These are obtained with a standard hole punch of science, so you just punch out small bits of leaf tissue and line them up here on the dish. You then put them in the presence of this bacteria that has your new DNA of interest. In this case is Agrobacterium tumefaciens. It's a bacteria that normally lives in soil and under natural conditions will transfer DNA from its own genome into the tree or into the plant, and then these will be stably incorporated. And what scientists have done is taken advantage of this bacterial trait and gotten the bacteria to transfer in the genes that we want it to transfer instead of its own genes. So that's very handy. I also wanted to mention here in the Strauss lab we're very much into reduce, reuse, recycle. So we have our lovely blob of recycled, reused tinfoil on here on the culture. So, good for the planet, right? And so then what we do once we put these two organisms together, our trees and our bacteria, is we then put the plant bits in the presence of naturally occurring plant hormones, and they'll actually divide and form new tiny little trees with the DNA of interest. And this is a great advantage of the amazing regenerative power of plants, and poplar trees here in particular, where you can grow an entire new tree starting with just a little tiny bit of tissue. So this is where we get our new research trees for genetic engineering. And I wanted to mention that the new DNA can encode a new trait of interest, say insect resistance, or it can alter a native gene, and I'll go into details on that later. And I wanted to mention that genetic engineering can add traits faster and more specifically than traditional breeding, but again, these are complementary approaches, so both can be used to obtain improved poplar trees. So just to go into some of the highlights of what we can do with genetic engineering, we do know that it can directly lead to biofuel benefits in poplar. So here are some examples of two recent publications. One showing that you can actually alter the cell wall composition in poplar, and this improves the efficiency of bioethanol production, so the trees have altered digestibility. Also showing another article here on the right. This came out of the Strauss lab, and it's where we created some actually herbicide-resistant poplars, so you can alter the weed management strategy so there's less competition, and you can actually get an improved yield from your trees. So these would be two direct traits of interest. We've also in the past used genetic engineering to put in insect resistance into poplar, so one of the stresses I mentioned before was insect pests. So you can see the tree on the left here has the insect resistance gene, and you can see the leaves at the top have a little tiny bit of nibbling by insects, but in general it's fairly undamaged, whereas the standard tree on the right, which is not resistant, looks a bit like Swiss cheese. Basically, cottonwood leaf beetle has come and chewed through a whole bunch of the leaves, so this actually can reduce the yield because you're getting the biomass nibbled up and the trees are stressed. And this could also open up the trees to susceptibility for other pathogens or pests to come and get a toehold, and again reduce the yield from your plantation. Some other traits that you can do with genetic engineering, you can create specialty chemicals. So I want to highlight a little bit of work here on work done at Washington State University on transgenic poplar producing some specialty chemicals. This is phenylethanol, chemical with a lot of uses in the cosmetic industry and the biofuel industry, and it's a very high value compound. It's shown here on the right, nice elegant little chemical structure here, and a nice phenyl ring, and then a couple of other carbon molecules afterwards. And this has a very floral scent, and it's normally found in roses and other flowers. And what the researchers have done is added the genes needed for the synthesis of this compound into poplar trees. So this is something that could be extracted from the leaves, and so be a very, very high valuable commodity trait. So this would be something very useful. Similar situation, we can also use genetic engineering to create another valuable co-product. So here we can put in 3 genes that are normally found in bacteria and put them into poplar, and you can form the precursor for biodegradable plastics. So this will be formed directly in the trees. This is a small polymer shown here on the left, and this could be something used to then create a biodegradable plastic. Currently, the methods used to create and express these genes, you have a trade-off between polymer production and plant growth. You're basically diverting carbon from the normal biomass pathway into this polymer pathway, and so the more polymer you make, the smaller the trees would be. You wouldn't get as much growth. So there is a balancing act here. So there is interest in refining this pathway to perhaps get a better yield. I just wanted to show you some of the possibilities that are out there. So we know we can get better trees through different methods. So I mentioned before there are some regulatory and social concerns, especially for the use of genetically engineered trees. So what are some of the concerns here? So the public in general, it can be worried about the use of genetically engineered organisms and genetically engineered trees. Some of that comes as carryover from concern over genetically engineered crops, which is something that's a little more familiar and widespread. Genetically engineered trees are more of a niche market. But here's an article I want to show you. This is a survey of people's opinions on the use of transgenic forest trees, and this was a survey that took place in Europe, which in general is a little more conservative considering genetic technologies. And one of the biggest concerns people had was they were thinking that if we use genetically engineered trees, there'd be loss of biodiversity. And one of the concerns was the risk of gene flow, so the idea that you'd have genes moving between the transgenic and the wild trees, and so that was considered a safety issue. So what is this gene flow that I've been mentioning earlier? So it's basically the movement of pollen and or seeds, or indeed entire organisms, from one population to another. So if you have a population here on the left, population 1, which would be your plantation, you could get the movement of pollen and seeds on the wind, which would then lead to the establishment of new individuals in population 2, for example here a wild forest. I should also mention at this point that you can get the movement of vegetative structures, so poplar trees can resprout. You could also get the movement possibly of vegetative structures, say on water, if a branch were to break off of a tree, float downriver, and then root in a bank, although in general vegetative spread is very, very local, slower, and could be controlled generally through standard propagation and management strategies. So that's more of a localized concern. But again, you can get movement of pollen and seeds on the wind, and this can be a very, very long distance, so this is a concern that people have. And so some of the ideas that you would be possibly quote-unquote contaminating the natural forest nearby if you had genes moving from your plantations outside the plantations. So this is one thought. Also wanted to briefly mention that there is concern in general over the use of genetically engineered trees, specifically if we were to use perhaps Roundup resistant or herbicide resistant trees, because we know from crops there was a very massive uptake and usage of herbicide resistant crops, which with the management strategies that were used at the time unfortunately led to the resistant populations of weeds coming up, where basically if all you use is one herbicide all the time on all of your weeds, you're selecting very strongly for herbicide resistant weeds. So we'd want to avoid any similar problems with any potentially future use of genetically engineered trees. So that's just something to keep in mind. I did want to say again that the hardwood biofuel plantation trees are not genetically engineered. They are hybrids though, often between exotic species. So I just want to show you a little bit about some of the different types of poplar trees that are available, some of which are in the hardwood biofuel plantations as hybrids. So here we have Populus nigra, that's usually found in Europe. It can be crossed with other species of poplars, such as Populus maximowitzi, which is found in Eastern Asia. We also have the Populus deltoides, which is found generally in the eastern United States, and all of these can be crossed together with each other. And we also have our northwestern variety, Populus trichocarpa. And so even though these poplar trees are generally found in different places or indeed different continents, they can be bred together and get these amazing hybrid offspring individuals. But again, they're often from these exotic locations. So there is concern over the potential impacts of exotic tree species. Could these trees come and become invasive in our environment? So the Forest Stewardship Council has a quote here that they're saying that the use of exotic species should be very carefully controlled and actively monitored to avoid adverse ecological impacts. So even though the trees are from different wild populations, since they are from exotic locations around the globe, there is concern over their possible use. And I did want to say that traditional breeding has not yet been able to find a solution to the issue of having trees that could spread either through sexual or vegetative means, although genetic engineering can be used to prevent the spread of pollen and or seeds. So I just want to mention that. So in the Strauss lab, we're thinking that genetic containment could be a possible solution to prevent the spread of genetically engineered trees or exotic tree species. So our goal is to use the power of genetic engineering to create non-flowering trees, or to create trees that have flowers that don't produce viable pollen or don't produce viable seed. So this would be something that would then prevent the spread as far as the pollen and seed movement, which again can go for miles and miles on the wind. And so since it can go for such a long distance, this could impact across many different boundaries. So you could imagine it crossing from one plantation to another, from one landowner's plantation to a neighbor's plantation, perhaps to a nearby forest. So there's quite a range of distance that could be potentially impacted. And what you're looking at here on the left is a picture of some of the poplar seeds, and so the little small oblong objects here inside of all this cotton-like fluff. And so they're very mobile on the wind, so they really can travel quite a ways. You might see them in the spring floating on the breeze. And so what we're doing in the Strauss lab is we're trying to target some of the fundamental fundamental regulators of flowers, so floral production and floral morphology, and we're doing this by the technique of RNA interference, otherwise known as RNAi. So for many decades now, scientists have been studying how do you create a flower. So what are the genes involved, which ones are critical. And so what you're looking at here on the top are two flowers from the model plant species of Arabidopsis thaliana. So a standard flower is shown on the left, so it has 4 white petals, and then a whole bunch of yellow anthers on the inside and 1 carpel. And so the anthers and the carpel are where the pollen and the seeds are formed. And if you see the flower on the right, you'll notice it looks very different. Instead of having the anthers and the carpel, what you see are a whole bunch of additional petals. And so this is a flower where the fundamental floral regulator, agamus, has been disrupted, and so now instead of getting those reproductive floral organs, you instead get a whole bunch of petals. And so this is one of the key flowering genes. And if you see here on the bottom, we have the same situation going on but in domestic apple trees. Since this was some work here in the Strauss lab along with partners at Cornell, where a standard apple flower on the left, and then an apple flower on the right, where we've targeted that same key fundamental floral regulator, agamus, and we're seeing a similar transition in floral form, where you're getting the loss of the pollen and the loss of the seeds. And so we're also doing the same approach in poplar. Now poplar doesn't have these typical flowers with the beautiful petals. I mean, we think they're beautiful, but we work on poplar, so of course we think they're lovely. But just want to mention that we're doing the same sort of gene targeting and same sort of method, but in poplar trees as well. And since we know that these genes are very, very conserved, we can feel confident that we can disrupt them and get the outcome that we're hoping for. So the method we're using to disrupt these genes is RNAi, this RNA interference. And we know that this technique can be used to produce diverse traits. Back in 2006, this method was actually awarded the Nobel Prize. It's been used to create a whole bunch of different trait modifications. I'm just showing a couple of nice examples here. So the large picture on the left is of a papaya plantation, where papaya trees were being decimated by this ring spot virus, which basically destroy the trees, destroy the yield. And so scientists have come up with a way to make a resistant variety. And so you can see the larger trees here are the ones that are, through genetic engineering, are resistant to the virus and actually grow, and they're healthy. Whereas the ones that are very tiny are the susceptible non-engineered varieties, and so you can see quite a distinct difference between them. And here on the right, just want to highlight a couple other examples. So you can use RNAi or RNAi-like techniques to create plum trees that are resistant to viruses as well. You can create apples that are resistant to browning, so a lot of food waste is due to products that either brown or are damaged in transport or other points in the processing. So these are non-browning apple slices. You can get a similar outcome with potatoes. So here we have potatoes that are they don't brown or turn spotty when you turn them into chips. So this is a useful trait through the power of genetic engineering. You can also get alterations in product quality as far as oil content. So just showing differences in different soybean oils, altering the oil composition. So what do we know so far? So I'm going to go through just a couple small highlights of our data from our poplar work here. So for the Strauss lab, we have a field trial where we're testing a bunch of our RNAi trees. So again, this is targeting those fundamental floral regulators. And we test them under field conditions. So this is a picture from last summer showing one of our students standing out in the trees. And she's standing near 2 different varieties. The ones on the left are Populus alba, and then the ones on the right are actually a hybrid. And these are looking to see, do we get effective reductions in flowering? Do we get healthy trees? We measure the size of the trees, we look at their leaves, we look to see if there are any massive alterations in tree form, we see if they're healthy, if they survive under tree conditions. And since we have quite a few of these trees, it's a very large undertaking, so field season actually starts about now for us and is ongoing through the summer. So it's really nice to be out in the great outdoors. And I want to show a highlight of some of our work working with RNAi interference against the key floral regulator leafy, or LFY for short, which in our field trial actually led to female sterile trees. And so then this here is just the highlight of the actual paper itself. And there's also a Harvard Biofuels newsletter if you want to read a little bit more. There's a hyperlink below to get to that article to summarize it. And so what do we see with our trees? So as I said, the trees appear to be female sterile. So if we go out to our plantation in March, when the flowers are starting to come out, what we see with our control trees are we're getting these beautiful catkin flowers. And in March, they're just starting to come out and form. And if you look at the flowers in the RNA leafy trees, they're tiny. We thought, well that's different. They're really small. And they didn't appear to have any of the carpel structures, which is where the seeds are formed in. And so we monitored these flowers over time. So if you go out again in April, you see that the standard wild-type trees have very, very large flowers. Each one of those small, kind of green bean-looking structures is a carpel, and it can have up to 4 seeds in it. And if you look at our RNA leafy trees, they're still small. So it's not just they're late bloomers and that they would maybe catch up later with more time. You can see they're still tiny, and they still don't have any carpels. And if you look in May, our control trees are forming that cotton fluff that I mentioned earlier, and they'll form seeds. Whereas the RNA leafy trees, the flowers don't really do anything. At this point, they basically dry up and fall off. And so the flowers that they do form, they're very tiny, they don't enlarge, and they don't make any seeds. So this is exactly something we're hoping to get. These are seedless trees, so they would be considered sexually sterile. So the question you're probably wondering is, well, the flowers look tiny, how does the rest of the tree look? So if you take a look at the trees here, they seem to have normal vegetative performance. So if you see one of the control trees here on the left and then the leafy tree here on the right, they look very, very similar. You might notice a slight difference in the branching pattern. The RNA leafy trees perhaps appear a little bit bushier, a little bit branchier. This isn't something we formally quantified, but something we'd be interested in perhaps investigating further. But what we did measure would be the tree height and then the tree diameter of the trunk, and we're not seeing any differences there. So they're putting on biomass quite well, they're growing quite tall, and their survival is identical. We also looked at a lot of the leaf traits. We're thinking that leaves would be a very sensitive output for tree health, that if the trees were stressed, perhaps the leaves would show some alterations. And we didn't see any major changes. We looked at leaf size, we looked at the amount of chlorophyll, so the green pigment in them. We looked at the leaf shape, we looked at the how much the leaves weigh, how dense are they, and we didn't see any significant differences between the two sets of trees. So it appears that vegetatively, at least so far, they appear to be pretty normal. So that's great. We're getting healthy trees, they're growing well. So this is something we're hoping to get, because there wouldn't be much point in having a sterile tree if it didn't also perform well. And I do want to mention that these sterile trees can still be propagated through standard vegetative tree propagation means, through vegetative cuttings. So typically when plantation trees are planted and created, they're not done through seeds, they're done through vegetative cuttings. We collect dormant branches and then dip them in some rooting hormone and then get a whole bunch of nice, identical, healthy cuttings. So this is exactly what we did with our trees, and so these are nice, healthy new trees starting by rooting branches from our sterile trees, and they grow well, they look great. So I just want to mention that just because a tree is sterile doesn't mean it's a dead end. You can still get new trees from that existing trees. You wouldn't be able to breed with that tree anymore at this point, but you would still be able to propagate it. Also wanted to mention that for a new research project in the lab, we're also trying the latest and greatest and amazing method for targeting floral genes. This is something in theory could be used to target any gene in any organism, and it's this CRISPR-Cas custom DNA modifying or gene editing technology. So this is actually the Science Magazine Breakthrough of the Year back for 2015. So it's really exciting because it's a way that we didn't have before to very precisely change single genes. And so we're using this to actually target those key floral regulators that I mentioned before, leafy and agamus. And for us, it's working quite well. So we're getting effective gene targeting, we're getting healthy trees. And so we're really excited by this because this is something that wasn't possible until just a few years ago. It's really a powerful new tool for scientists. So we're very excited about the possibilities. And as an interesting nuance, this method can actually be used to create organisms that are not considered as genetically engineered. So there are ways to apply this technology that don't lead to a what's considered to be a genetically engineered product in the end. So just here showing an example from mushrooms, where these ones have a precise change to a single mushroom gene. And this change is done where the CRISPR DNA itself, no extra genes or DNA are present, just the change. And it's targeting a single gene in the mushrooms to make it so they don't brown anymore when you cut them. So this would be a very useful trait as far as product quality. And so the thought is one could possibly do a similar approach in trees, that you could just have trees with only the change but without any extra DNA. And so a similar application might be acceptable by certification systems or by other regulators. This is just another option that's on the horizon, very near future, where one could possibly have this approach as well. So very excited by that. Advance the slides here. Oh, there we go. So what's coming up next? So what's new in the Strauss lab? So one thing that we're doing is actually scaling up a larger field test for RNA leafy trees. We want to get a better handle on the vegetative performance and survival and long term. This would be the next step in the test. You always want to be able to replicate your results. And then something we're very excited about that's coming up right now is we're setting up to do some greenhouse and field tests of trees with those floral genes targeted by that CRISPR technology. So this here would be an example of some little tiny CRISPR trees in a box that are getting ready for our greenhouse and for our field studies. So that's something that's coming up. So we'll be looking at their survival, their size, the effectiveness of the targeting on the floral form. So this is something that we're working on right now. So latest and greatest. So what does the future hold? What are some of the prospects? So we think that the future use of genetic containment technologies and poplars could be very beneficial for biofuel plantations, such as the one shown here below. So I just want to reiterate again, all AHB field sites are non-GE, they're hybrids of poplar species. But if the exotic species, which they are, becomes a concern, or if genetically engineered varieties were tested, then we think that our containment technologies could be used to prevent flowering, especially if any fields have earlier flowering. So they are normally harvested on a 3-year cycle, but if a field isn't harvested as planned, or if some shoots were missed, or if a plantation underwent, say, a change in ownership, so there was a delay in the harvesting, then this containment could be used to prevent flowering if needed. It could also prevent the movement of pollen and seeds to wild populations, again to feral trees or other plantations. So I think it could be great enabling technology to facilitate the use of GE or hybrid exotics. The future prospects. So we know that genetic engineering can provide considerable value and considerable benefits to poplar plantations, and some of those benefits are directly related to hardwood biofuel production. Right now there are obstacles to commercial use, so we cannot commercially use genetically engineered trees at this time. Again, the main concern for folks is the gene flow, the thought that these genetically engineered genes could move out of the plantation. But we know that sterility could be a solution, so this genetic containment technology could be a way to prevent that gene flow, and we know that it works under field conditions. We know that new developments in gene editing technology, so this CRISPR, would also be another possibility for tree containment and might be another way to obtain the traits of interest without them trees being considered genetically engineered as well. So many, many exciting possibilities on the horizon. So I just wanted to finish up by acknowledging the Strauss Lab members who are directly involved in this work, especially Kathleen Ma, who's our tissue culture and transformation guru, been at the Strauss Lab a number of years. Also Steph Lariaga, who's been doing the CRISPR-related mutagenesis work with the poplar trees. And also our field managers, past and present, so currently Anna Magnuson and previously Corey Alt, as well as the funding agencies, particularly Advanced Hybrid Biofuels Northwest, the United States Department of Agriculture, and the industrial members of our research co-op here at Oregon State University. Thank you, and I'm very happy to answer questions people would have.
Thank you very much, Amy and Steve, who is there with you. We have had one question come in, and I'll just quickly read this off for everyone. Given the apparent polygenic nature of many of the desirable traits for growth, water use efficiency, stem form, wood chemistry, do you think useful genetic modification using the CRISPR system will be as productive as recurrent breeding and selection in identifying commercially successful varietals? So Steve and Amy, if you want to turn your mic back on and answer that question, we'd appreciate it.
I can, this is Steve Strauss, I can make a quick reply to that. 2 points. One is that, to reemphasize, we don't see CRISPR or any other GE approach being something that's exclusive to, is competitive with conventional breeding. You still need conventional breeding, using those polygenic kinds of traits as the foundation. But that also doesn't preclude you from making sort of step gains by emphasizing individual genes to promote any of the traits that were mentioned there. And this has been done in research. So we think both things are possible. You will use genetic engineering. The basis of those traits in nature is polygenic, but in genetic engineering, particularly as you help trees adapt to the novel systems of biofuel production, you can also make these step gains by adding these individual particular traits, either for quality or for productivity or for disease tolerance, as you saw some examples.
Steve and Amy, can you see the follow-up question there?
Yes.
It's a long one to read, right?
So I'll let you guys do it.
Yeah, we're looking at it right now and thinking about it. Okay. So the first point, that the trees in the ground that resprout are something that, from the point of view of regulated research, needs to be controlled, and that does produce challenges, just as it does to poplar growers who want to change from one genotype to another or change from poplars to a different crop. And so that is true, and that's something that we actively work with USDA under our permits to manage. And so that is just the truth. We can manage it, and we've been doing that for many, many years, but sometimes it definitely presents challenges because poplars are persistent, and that's definitely true. The question of whether there's a potential for lateral transfer of the trait through agrobacterium, I don't believe there's examples in sort of what I call ecological time of agrobacterium picking up a trait and transferring it. I think over evolutionary time, millions of years, those kinds of things can happen, but the frequency of that happening in our lifetimes in a scale of tens to hundreds of years would be extremely small. So I think those would probably be something you would recognize as a legitimate concern and possibility, but the literature would say that it's extremely improbable. Thank you very much, Jeff. Good challenging questions.
Very thought-provoking.
So we actually have a question for you guys here in the Biofuels office. Patricia has a question for you.
Yes, thank you, Amy and Steve, for an interesting webinar today. Appreciate that. This is something that you mentioned during the webinar, and some of you may be aware that EPA is under doing proposed rule changes for poplar as a biofuel feedstock. And some of the questions we have been asked is about the concern over invasive potential. And I know, Amy, you mentioned the vegetative spread concern, that it was minimal, but if you're harvesting trees, with this short rotation methodology on a 2 to 3 years, and you're chopping up the wood, it does seem feasible that cuttings or something could spread. And I just wanted to know your thoughts on that. Even if you have cuttings, does the fact that they are sterile, does that minimize any potential concern that they could spread? So even if a cutting did spread, it's going to be a sterile tree, and so it's not going to be contaminating natural populations like it would if it was not a sterile tree.
So I did want to mention that so our trees can still spread vegetatively, as you saw. Vegetative spread is something that generally should be very local, so that'd be something managed more at the plantation scale. So, I mean, plantation owners aren't going to want random trees popping up all over the place. So normally, in our plantations, we look for any suckers or any unwanted trees, and those are removed. You're not wanting to have uncontrolled vegetative spread in general. Since the vegetative propagation seems to be more local, that's something that should be able to be contained with standard management practices, so you shouldn't be getting any high frequency of vegetative spread even on a site basis. So yes, there is the low risk possibility of cuttings taking root in unwanted places, but again, we think that's something that's relatively low frequency in relatively short distance. So our strategy, again, will only really prevent pollen and seeds and won't do anything for the vegetative spread, but again, we think that's something that can be managed at the local level.
Yeah, just to add a little bit more to that, you know, it's definitely not a zero possibility, as Amy said, and it's something if you had a trait that was of concern, you'd want to monitor for it. And indeed, many companies now around the world that plant exotics where they're of concern to certification, our government agencies have to monitor themselves around the plantation, in the plantation, so there isn't spread. So that's a possibility. And the contrast is that to the potential of seed and pollen, which, as Amy said, can move, you know, hundreds of meters to many kilometers. So very, very, very rapidly, the spread is orders of magnitude greater and really beyond the ability of anybody to manage it locally. So that's a dramatically different situation. And we could, in the future, try to engineer vegetative propagation so that would not happen. But that would be another step of complexity that I would see as a second generation of engineering effort.
Right, so thank you again to Amy and Steve. If you guys have more questions, please feel free to enter them into the chat window. Really quick, I wanted to mention some of the upcoming events that we have going on, and we do have a couple of more poll questions that we'd really appreciate if you could answer. This helps us evaluate the usefulness of our webinars. So next month, we're going to hear from Nora Hayter, who is one of our colleagues at the Extension Office, and then also Peter Moulton, who is on the AHB Advisory Board. He works with Washington State Department of Commerce. And they're going to talk about policymakers in the Pacific Northwest and how we as a project, for example, can communicate with them, how the policymaking system works. And we actually did a survey at the Extension Office, and Nora will talk about those results. In March, we're hoping to do a webinar that's focused on the students at University of Washington, and we'll have a series of short presentations to highlight what the conversion graduate students have been up to. We're very excited to be able to host field tours of poplar harvests. We're still waiting to know exactly when those harvests will happen, but that will happen sometime probably in February or March of this year. We'll be harvesting in Hayden, Idaho, and then also Jefferson, Oregon. If you want to know more about these events or be updated when we have more information, please visit our website, hardwoodbiofuels.org/events, and you can register, and we will send emails as we learn things. So I see we have a few of the poll questions being answered. If we could have more people answer those would be great. And again, if there are any last-minute questions, please type those into the chat box.
Thank you very much, folks, for listening in. It's been really fun presenting to you. And thank you to the folks at the Extension Office for giving us the opportunity to speak to you today. It's been really enjoyable.
Well, thank you everyone for joining us. I will leave the webinar up just for a little bit longer so people can answer poll questions. And feel free to contact me at noel.hart@wsu.edu if you have questions, if you want me to send you the link to access the webinar later, or if you need the presentation, I can get in touch with Amy and Steve for you. Thank you, everyone. Have a good day, and stay warm if you're in the colder, snowy portions of the U.S.